recording really quick. Our usual set of announcements. Just for the Q&A, we are going to be doing that at the end of the talk. So if you have questions, please feel free to drop them in the chat, or you can use the raise hand function to ask them at the end of the day, or not at the end of the day, at the end of the talk. Yep, if you have any other unrelated questions, feel free to message me directly. Paul will be putting the attendance link into the chat. So if you are enrolled in either the MolES or chemistry courses, please be sure to go there and sign up for those. A special welcome to our guests here that are not part of those courses. This is our third talk in our Science of COVID-19 seminar series, so thank you for joining us. Hope that you enjoy it. We're really excited for today's seminar, and I will now introduce our speaker, who's Dr. David Baker. David Baker is the director of the Institute for Protein Design. He is a Howard Hughes Medical Institute investigator, a professor of biochemistry, and an adjunct professor of genome sciences.
No, nor mine.
Oh, sorry, somebody is unmuted here. If you could all make sure that we're muted, that would be great. All right, thank you. Sorry, he is an adjunct professor of genome sciences, bioengineering, chemical engineering, computer science, and physics at the University of Washington. His research group is a world leader in protein design and protein structure prediction. He received his PhD in biochemistry with Randy Sheckman at UC Berkeley and did postdoctoral work in biophysics with David Agard at UCSF. Dr. Baker is a member of the National Academy of Sciences and the American Academy of Arts and Sciences. He is a recipient of the Breakthrough Prize in Life Sciences, Irving Segal and Hans Neurath Awards from the Protein Society, the Overton Prize from the ISCB, the Feynman Prize from the Foresight Institute, the AAAS Newcomb Cleveland Prize, the Sackler Prize in Biophysics, and the Centenary Award from the Biochemical Society. He's also received awards from the National Science Foundation, the Beckman Foundation, and the Packard Foundation. Dr. Baker has published over 500 research papers and been granted over 100 patents and co-founded 11 companies. 75 of his mentees have gone on to independent faculty positions as well. So as you can tell, it is quite the resume. So we're very excited to have Dr. Baker speaking with us today, and I will go ahead and hand it over to him. Welcome, Dr. Baker.
All right, well, thank you. Yeah, it's always fun to speak here, and if you stuff that you're interested in, you can follow up with me, or you can follow up with the people actually doing the work, since we're all right here. So I'm going to talk about protein design today, and with sort of an emphasis on COVID at the beginning. And the approach that we've taken to protein design up until very recently was sort of a physically based approach, where the design work is based on the hypothesis that the folded states of proteins are likely global energy minima for their sequences. So what this means is that if you want to predict the structure of a protein from its amino acid sequence, you have to search through the different possible states of the protein chain and identify the one that has lowest energy. If you want to design a new protein, then you need, once you've decided on the structure you want to design, then you need to find a sequence which has the property that its lowest energy state is that structure. And so to do these calculations, you need to be able to calculate the energies of different configurations of a protein molecule, and you need to be able to sample through the possible sequences and the possible structures. And our colleagues have developed a program called Rosetta over the years to do these calculations. Now, what I'm going to come back to at the end of my talk is a completely different picture for designing proteins, which doesn't really involve energy calculations at all. Instead, it involves identifying sort of recurring features of proteins and their sequences. And that's a deep learning approach for protein design. And so that'll be at the end. But until then, everything I'm going to describe to you is designed in this way. So we decide on a structure we want to make. We identify a sequence whose lowest energy state is that structure, and then we make the sequence. By making the sequence, what I mean is that we synthesize a gene that encodes this new protein, and then we put that gene into bacteria, and we make the protein in in bacterial cells or yeast cells or mammalian cells. And then we get the protein out and we determine whether it has the structure and function that we designed. So I'm going to, my research group is really focused on the problem of de novo protein design. And this is an interesting problem because the space of possible proteins is really enormous. So with 20 possible amino acids at each position, and that's really an underestimate because there's a large number of possible unnatural amino acids. But even with just 20 and 100 different positions in a protein, so here's a protein chain, first residue, second residue, third residue, fourth residue, so forth. The number of possible states is 20 times 20 times 20, 100 times, which is a number around 10 to 130th, which is really a huge number. In contrast, the number of naturally occurring proteins is many, many orders of magnitude smaller. And moreover, naturally occurring proteins sort of cluster into families. And so, so there are, there's just huge amounts of unexplored space. So most protein engineering to date has involved taking naturally occurring proteins and modifying them a little bit to modify their function. And instead, what I'm going to tell you about today is all de novo protein design. So we're building proteins from scratch from the sort of first principles I described just on my preceding slide to have new functions. And I'm going to illustrate protein design on with the work we've been doing against COVID. And the when COVID came along, we were sort of caught by surprise, like everybody else. And but we were able to take the protein design technology we've been developing for the last recent years and apply it to COVID. And it's been really exciting to be trying to do something that could really make a difference in the pandemic. So I'm going to describe Antivirals Diagnostics and briefly say a few words about vaccines. So once we have this, the sequence, the genome sequence of the virus, we could predict the structure. And once we had that predicted structure, we could start doing calculations on it. Likewise, we could, so when the cryo-EM structures and crystal structures became available, we switched to using those. So I'm going to first talk about designing antivirals. So these are proteins that are going to, once you've gotten infected, and these proteins are going to help you get rid of the virus. And we focused on this interaction between the ACE2 receptor and the spike protein of the virus. And so this is the human cell, this is the coronavirus, and this is how it gets in. And so we wanted to make a proteins that would bind to the receptor binding domain and prevent it from interacting with the ACE2 receptor. So we took two approaches. One was to take the alpha helix in ACE2 that forms the majority of the binding site with the RBD and build a small protein around it, shown here. The second approach was to create very large libraries of sort of made up proteins and dock them against the RBD and identify those that are the most shape complementary, and then redesign the interface between the protein and the target. So in both cases, we had obtained many. We got from these calculations, we got many designs. Each one corresponds to has a particular amino acid sequence. We encode those in synthetic genes, and we actually screened very large numbers of designs, taking advantage of sort of modern chip-based gene synthesis, which lets you encode very large numbers of brand new sequences in DNA. And we identified those that bound the RBD, the spike protein. And here's what two of them look like. This protein is the one that's based on ACE2, and it's 75 amino acids. This one is based on, this is just a brand new made up protein. It's 55 amino acids, and it's a very, very stable protein. Now, you might think it would actually be easier to design by kind of stealing something from a naturally occurring protein. But actually, this protein here binds with about 10 nanomolar affinity, whereas this one binds about 100 times more tightly at about 100 picomolar, It's kind of a less constrained optimization here, because this protein can bind to the virus in any way that it has there was more room for optimization, because we weren't confined to binding in the same way the ACE2 receptor does. So this protein binds the target about as tightly as ACE2 does. Okay, so with David Wiesler and Lexi Walls here at the UW, I should say that this design work, the work in designing these two proteins, was done by Longjin Cao, a postdoc in the lab, working with Brian Coventry, who just graduated, got his PhD from the molecular engineering program. And they had made really quite a breakthrough in design of protein interfaces in the 6 month leading up preceding COVID. And so they were able to take the techniques that they had developed and apply them to this challenge. So here's a picture of the protein that was on the right, the little 55 residue protein, and you can see there are 3 copies bound to the spike protein. This is a cryo-EM structure, and it's really neat that the way that this small protein binds to the RBD is nearly identical to the computational model. So there's a superposition of the design structure in the computational model, and that holds even at the level of side chains. So these calculations, we're making up these brand new proteins from scratch, and they can bind in exactly as they were designed to. Now, many of our designs didn't work at all, didn't bind at all. They probably don't even fold. But so the calculations are imperfect, but when we do get it right, we get it right for the right reason. So we have a lot of data now on showing that these proteins protect cells and animals from the virus. So this is a first experiment where here virus particles are being added to cells, and we add in the varying amounts of the protein. And this magenta line corresponds to the protein on the preceding slide. So you can see about in the 15 picomolar range, we're getting inhibition of viral entry. So it's a very, very potent inhibitor of viral uptake. So we have quite a lot of data in mice and hamsters on this protein. So this is one example of an experiment where, in this case, this is the Syrian hamster model, which is the primary animal model for COVID. And they're given the protein, they're given it before 48 hours before they're infected with the virus to 24 hours after. And then there was one group of animals that didn't get the protein at all. And you can see that in this period, minus 48 hours to plus 24 hours, there is, the animals are substantially protected from the virus. Now, you all have heard about the escape variants that are coming up. And so we wanted to our goal has been to make single compounds that were basically escape resistant. And as I showed you before, the spike trimer binds 3 copies of these domains. We have actually a number of different domains that bind in different places. So in order to obtain escape resistance, we've been connecting these with flexible linkers. And just to show you the rather remarkable things that happen when you do this, here's an example of an experiment. This is from Andrew Hunt at Northwestern, where he took the monomeric proteins that I told you about. This is the ACE2 mimic. This is the one the 15 picomolar one. And if you just add them to the virus and you wait several days and you measure how much protein it remains bound, so the kind of an off-rate experiment, you find that basically it almost all dissociates. But if you link these proteins together, then the off rate goes way down, and you can see that even after 14 days, the majority of the protein is bound, which is very encouraging. So we can make things that essentially don't come off the virus. And so then with Brett Case in Mike Diamond's lab at WashU, he took some of these constructs, and they're shown here. So they have these 3 domains arranged in different formats. Here is a trimeric one with 3 copies of the same domain, And here's 3 different domains linked together by linkers. You can see sorry, this slide got a little messed up, but this is the South Africa strain of the virus, the UK strain, and the Brazil strain. And you see these 3 compounds are effectively neutralizing all 3 strains of the all 3 escape mutant strains really quite potently, essentially as potently as we were neutralizing the original strain with the single monomer. So we're really excited about these. So it's not I don't believe, I mean, our collaborators are not aware of any other single compounds that can neutralize all of the different forms of the virus that have been found to date. Um, so um, so another nice thing about these proteins, antibodies are generally one of the most widely used sort of common therapeutic proteins, but they're expensive to make. You have to make them in 1 million cells. Whereas these proteins, this is just the monomer, expresses at very high level in cells. It's this 55 residue band at the bottom. And then when you heat the cells, most of the proteins are insoluble. And what's left in after you heat the cells, is just mainly just the protein in solution. So we think we could make this protein. These proteins can be made extremely cheaply. And so what we're imagining is, what we're thinking about, and actually we're proceeding really rapidly towards, is a nasal spray that you would take before infection if you're going into a dangerous area, or immediately, or within the first couple of days after infection when you have symptoms, and that this thing we think we can make really, really cheaply. And we actually are now proceeding towards clinical trials for this with a pharma partner, and that will start in the fall, which we're very excited about. So the second area I wanted to talk about is diagnostics. And here, in addition to designing proteins that bind very tightly to other proteins, we've also been designing proteins that are essentially molecular devices that have, here's a system with 2 states. It has an off state and an on state. And the system's set up so in the off state, it's dark, but in the on state, it emits light because a luciferase enzyme is reconstituted. And we can couple the opening of this, going from the off state to the on state, to binding of a target. And in this case, to make a coronavirus sensor, we took the same small protein that I showed you before, which is now embedded in the sensor. So in the off state, in the absence of target, it's completely dark. And then, but in the presence of viral proteins, either the spike or the RBD, you get this rapid increase in the amount of light emitted. You can read this out on a plate or on paper, and with a cell phone. So we are excited about this. So what we are focusing on now is developing this as a way for individuals to monitor what their level of neutralizing antibody is. So as I told you, we have this is basically the off state. The protein comes in, and I told you that it's thermodynamically coupled, so when the protein's present, the switch opens. And now, if you add in antibodies which are neutralizing and bind at the ACE2 binding site, you pull off the RBD. And I told you this is a reversible system, so you lose signal. So the amount of signal you lose depends on how much antibody is there that binds at that site. And so we can use this change in luminescence to read out neutralizing responses. And here's some experimental data. My colleague Neil King, I'll say in a few words in a minute, has been developing very promising vaccines for the virus. And you take mice that have not seen the vaccine, and you take their sera, they make essentially no antibody response to the, and there's no change in the luminescence of the sensor. But as the mice are vaccinated, they make increasing amounts of antibodies against the virus, and that we can detect with the sensor in the way I described. And so here's the change in luminescence versus the neutralizing titer in the sera. So we're now trying to get this out in the world. Basically, again, it would be a very simple home test that you can read with a cell phone that would tell you the extent of neutralizing antibodies you have. So at different periods. So if you've just gotten vaccinated and you're wondering how long it takes for your immune system to ramp up, or you want to know, it's been 6 months or a year and you want to know whether you're still protected, you can use this kind of test. And you could also use it to figure out what strains you're protected against. And we can use this molecular sensing system to detect a wide range of different compounds. For example, here's a sensor for troponin I, which is a marker of heart disease. And here are sensors directly for a few of the SARS antibodies. And the final area that I've talked about the antivirals, the diagnostics. The final area I'll talk about just very briefly is vaccines. So, another example of the sorts of things we've been designing are self-assembling nanostructures, like these self-assembling two-component icosahedral particles. And this one's made of a pentamer and trimer that have interfaces designed in them that direct assembly of this particle here. And this is what they look like. They're very homogeneous particles. So my colleague Neil King and his group have, along with David Wiesler at the UW and others, have taken the RBD for the spike RBD, the same protein I've told you about, and put that on the outside of these nanoparticles. And what they found is when they put these into animals, they elicit really, really strong immune responses, and much stronger than with the current generation of sort of trimer-based vaccines. And we're excited because those compounds, those vaccine candidates, have actually gone into people starting about a month ago, and as part of a phase 1, phase 2 trial. So overall, it's pretty exciting. We have all these kind of crazy methods we've been developing that are actually creating, antivirals and vaccines and diagnostics are actually getting out in the world, which is really, really neat to see. Okay, so now I'm going to leave COVID behind and tell you about some of the more general work that we have been doing in the group. And one is following up on this idea of nano cages. So what I described before were two proteins that we designed an interface between that then self-assembled into a nanoparticle. But what we realized is that we could do the same thing, but require that one of the proteins have a function. And the function we chose was antibodies. So what we did is we designed proteins that, for example, in this case, a pentameric protein that binds to antibody molecules in such a way that that it directs their assembly into these icosahedral structures shown here. So this is actually cryo-EM data of an antibody nanoparticle basically obtained by mixing essentially any antibody with an FC with the designed protein. And Robbie Devine, who's did this work, actually used this technique to make a wide variety of different antibody assembling proteins that assemble antibodies into, for example, these dihedral structures, tetrahedral structures where the antibodies come out at the corners of the tetrahedron, octahedrals where they're in the center of edges of squares, and here are two icosahedral designs. And these ones have 30 antibodies each. So Marty Tooley and Marty's a molecular engineering student, showed that if you assemble these sorts of particles, and you get basically a multivalent antibody nanoparticle, and these can have very potent effects on signaling. So this is a measure of activation of a B cell marker. And you can see that the particle itself is giving strong stimulation, whereas the antibody alone has essentially no effect. So we're very excited about this as a way of sort of generating agonists from any of the wide variety of antibodies that have been discovered. So on this topic of self-assembly, so far I've talked about the assembly of proteins in closed structures like icosahedra, but Ariel Ben-Sasson did similar things, designing interfaces between two proteins that direct the formation not of a closed structure, but a flat two-dimensional structure. And so here's sort of the design scheme shown here. The two proteins interface between them is designed, and you get it's supposed to form a hexagonal lattice. I have another picture here. So this is the design model in magenta and blue. And this is what you actually see when you take the two proteins, you make them in bacteria, and then you mix them together. You get this really extensive hexagonal lattice. And you can get a sense of the scale for it of it here. And these structures were determined by Matt Johnson and Justin Coleman's group. Now, these have very interesting effects on cells. If you attach to these subunits cellular receptor binding proteins, they actually bind to the cells, the arrays form, and then they block endocytosis because there's so much, because they kind of stiffen the membrane in the region. And over longer time scales, they self-assemble into really large clusters, which can direct cell polarity. So it's kind of really excited now about designing proteins that can sort of for a synthetic biology that involves cells and design proteins. So leaving the topic of nanostructures, now coming back, I'm going to come back a little bit to this design of multi-state systems that I talked about before. So I told you about the diagnostic where we had two components where the cage was closed until the other component came in. So we've used this technique to for the problem of being able to target cancer cells based on combinations of surface markers. So if you have a tumor that just has a single marker on its surface, it's pretty easy to detect it with an antibody. But if the tumor has two, it doesn't really differ from naturally occurring natural cells, normal cells, by the presence of any one marker, but has two markers that are unique. You need some way of doing AND logic on the surface of the cell. And the basic idea here is we can direct one of these components to one marker, the other component to the other marker, and we only get opening of the switch when both markers are present. And then we can couple opening of the switch to recruitment of an effector. And this is work by Marc Lajoie and Scott Boykin when they were in the lab, and Gillian Bruffy, who's a graduate student now. And in collaboration with Stan Riddell's lab, Alex Salter, we sort of went one step further. This is not just 1 and 2 logic, but it's 1 and 2, not 3. So imagine you have a tumor cell that has two markers that are associated with the tumor cell and one marker that's associated with a healthy cell. So you want to kill the cells that have markers 1 and 2, but not those that have marker 3. And so we can do it in the sort of way I described. We direct the two components of the switch to markers 1 and 2. And then to the third component, we target a third protein, which actually sequesters one of the components. And here are cells that are expressing either no components, either one component or the other, both together, or both together plus the healthy cell marker. And you can see we're only getting, in this case, what we're incorporating is something that recruits CAR T cells. And you can see we're only getting activation when the two markers are present, but not all three. Now, jumping to a different area, everything I've talked about so far is are soluble proteins, but we've been very interested in membrane proteins and membrane channels. And so this is work by Chun-Fu Zhu. He designed proteins that are cylindrically symmetric and have pores in the middle. This has a small pore about the size for a water molecule to get through, and this one has a larger pore. And here's another look at the smaller pore protein. Here we've designed, Pei Long and Chun-Fu redesigned the outside of this protein so it would insert into membranes. And Tamer and Bill Catterall's lab show that this protein is actually a very selective potassium channel. So here is, this shows you that the potassium is getting through these channels much more efficiently than sodium. So it's kind of interesting. This very simple design gives you a fairly selective potassium channel. And we can put cysteine up here and then modify it. And if we do that, the conductance completely goes away. So this is the unmodified protein, the protein modified with different reagents, suggesting that the ions are traversing the the central channel as in the design. And this is a crystal structure, cryo-EM structure from Pei Long of the larger pore. which is actually large enough to get small organic dyes and compounds that are about 1 kilodalton through. So those were transmembrane helical barrels, and Anastasia Vorobyeva decided to take on the challenge of designing outer membrane beta barrels. So these are beta barrels that span the membrane, and as many of you are aware, single molecule DNA sequencing methods like those pioneered by Jens Gundlach here at the UW rely on these sorts of proteins because they're very robust, and you can get them to insert post-translationally into membranes. So Anastasia worked out sort of general principles for designing this class of proteins. It was very interesting. A key thing turned out to be weakening the propensity of these strands, these beta strands to form. The problem is if they form too quickly, you get amyloid, and the proteins simply don't fold into monomeric states. And this shows a crystal structure of the design compared to the computational model. So Samuel Berhana and Yulai Liu in the group are now taking these methods and extending them to design proteins with large central pores, which we are then excited to start adapt to creating new types of filtration devices and perhaps new approaches to peptide and DNA sequencing, because we can now customize the inside of the pore really for those applications. So I just wanted to mention we also have a game that's and Fold It player called Fold It, and Fold It players are also having fun designing proteins, and this shows some of the really cool proteins that they're designing, and this is sort of what the game interface looks like. And Brian Koepnick actually made a whole bunch of these proteins that the Fold It players designed, and turned out they folded in the lab, which is really cool. A final example of sort of what we can do with more traditional protein design. This is work from Alexis Courbet, postdoc in the lab, working with Jesse Hansen on the cryo-EM with Justin Coleman here at the UW. So this is where the goal is to make molecular machines. And so Alexis designs these axles, which are shown here, and rings shown here. Then he can assemble the rings on the axles. And some examples of those are shown here. You can see you can see this is now cryo-EM data showing the ring on the axle. And you can see it's kind of blurry in the region of the ring. That's because there's different rotational states, because these rings can rotate along the axle. And this is a higher resolution structure here of one of these. And you can again see the arms of the rotor coming off the axle. And so now it's very exciting time to investigate the rotational landscape. And Alexei and postdoc in the lab, Yakov, are working to incorporate catalytic sites at the interface between the rotor and the axle to try and drive motion. And we have some very preliminary single molecule data with Zeb Bryant's group at Stanford suggesting that this might be happening. I think I will, just to summarize, I've shown you a lot of things. I think we understand enough about the fundamentals of protein folding and assembly to really build a whole new world of functional proteins. Let me acknowledge the people who've done the work, and then I'll take questions. And I think I might be running out of time. If people want to hear more about hear about deep learning, I can talk about that after questions for anyone who's interested. So I've had really amazing colleagues here. Longjing Cao, amazing, brilliant postdoc in the group, basically lived in the lab for three months when COVID broke out, designing, sort of experimentally testing those proteins. Worked very closely with Brian Coventry, who really, and the two of them really made a big breakthrough in designing protein-protein interactions. Ina Greshnik worked with them closely on the experimental side, and then we've had really outstanding collaborators for the mouse and hamster work. The diagnostic work of Alfredo Rubio and Andy Ye, and that's been very exciting. As I said, we're currently trying to get this out of the UW so we can get this into drugstores so people can use it. Neil King's group, I briefly alluded to the vaccines. That's all going on in Neil's group. The antibody Nano Cages was worked by graduate student Robbie Devine, and David Wiesler and Hash solved the structures I showed. The self-assembling 2D lattice, that was worked by Ariel Ben Sasson and the work on endocytosis and the effect and investigating the impact of these arrays on live cells, done by Joe Watson and Emmanuel Dury. This was actually published last month, so you can read much more about it. The designed helical membrane proteins and channels, work by Chun-Fu Zhu and Pei Long in collaboration with Bill Catterall's lab, and the beta barrels, work by Anastasia Vorobyeva, who now has her own position in Belgium, and Samuel and Yulai are continuing her work here in the lab. The design protein logic work done by Mark, Scott, and Jillian's now applying this to a number of, to both DNA delivery and cancer applications, and it was a close collaboration with Stan Riddell and Alex Salter. I just mentioned the rotary motor work of Alexei, and he's been working with Jesse. And I'll maybe say a few words about this if there's interest, but let me take a few questions first.
Awesome. Thank you so much, Dr. Baker. That was a wonderful talk. Just really cool. Just a reminder for all of our attendees here, if you would like to ask a question, you can either put them in the chat or you can use the raise hand function in the participants tab, and I will call on you and you can unmute and ask your question that way. And I guess to get us started, I am just curious about your thoughts, kind of going back to COVID. What are your thoughts about sort of like the nimbleness of the work that you're doing in terms of adapting as the virus, as like the new variants of the virus come out, and making like diagnostics or even vaccines for that?
Yeah, well, it's a very good question. And there's a bit of an arms race going on now, just generally with major vaccine manufacturers making a round two. What we're really hoping on, so Neil's already making vaccines for the new variants. We're hoping with the compounds that we're designing, the antivirals, that they will be essentially escape resistant. And we don't know if that will be possible, but they're binding so tightly and against all the escape variants thus far that we're reasonably optimistic. I think so as far as nimbleness goes, I think where we're really focusing is on sort of the next pandemic, and trying to work out how to improve these methods to the point where you could design really high affinity binders, in just a couple weeks after infection. So sort of the general methods for really streamlining things. And of course, with the current binders, if new variants do turn up that in fact do evade even these multivalent versions, then it should be, it'll be pretty straightforward to design compounds or modify the ones we have. The problem is that you have to go through the whole drug manufacturing thing again. So we're really hoping we can avoid that.
Yeah, thank you. That's great. All right, we have a question from Mehmet Sarikaya. He's asking, what is deep network hallucination?
Oh, well, I don't know. Should I just go through that? It'll be about 4 more minutes. I was just worried about running long.
Sure, we have plenty of time if you want to go for that.
Actually, what time is it? I lost track.
It is 1:40.
Okay, all right. So I'll just go through. Okay, so that was not a plan, but okay. So what is protein designed by deep network hallucination? So everything I told you about so far was based on these sort of energy-based calculations. We have basically an energy function, and you sum the hydrogen bonds and van der Waals interactions and electrostatic interactions and so forth. So, so now I'm going to talk about sort of a completely different way of doing protein design. So we and others over the past several years have been working on basically neural networks, deep neural networks that can take amino acid sequences and predict the distances between the different residues in the protein, and from that generate three-dimensional structures. So even Anashanka and Sergey Chinnikov realized that once you have such a network, you could use it to generate ideal proteins. And I think many of you are familiar with the idea of the fact that if you have a network that's trained, for example, on recognizing images of cats on the internet, so it's something that just classifies images as cats. You can then start generating random images and start optimizing them until the network starts thinking they're cats. And then you get images of cats coming out of the network, but they aren't any actual cat. They're just kind of an ideal version of a cat. And so similarly, here we can start with random sequences And then we predict their structure, and it doesn't really look like a protein at all, but we can then optimize the sequence so that it looks more and more like a protein. And so this is sort of an example of how this works. We start with a random sequence, and this is a contact map. So this is residue by residue, and this is showing you whether the residues are in contact. And then we can do this sort of sequence optimization. This is just straightforward Monte Carlo optimization. And at the end of the day, we get these very featured contact maps predicting lots of contacts between the amino acids. And then those correspond to the different folded structures. And this is so we can run this many, many times, and we generate all sorts of different proteins. And we can make them in the lab, and they are folded, which is pretty exciting. And we've been able to solve the structures of a couple of these, and they're very close to the design model. So we can make the protein is just coming up with these ideal things that look like ideal proteins, and we can make them in the lab. They actually fold into those structures. And the experimental work was done by Sam Pellick and Tamuka Chichukuri. So there are some neat things that happen here. I think one of the things that's kind of cool about this design process, it can be aware of the whole energy landscape, but because of time, maybe I'll skip through that to just a final couple slides here, which is we can, so if we freely hallucinate proteins, it's not, I mean, it's kind of cool. You make up these brand new proteins, but they don't really have any function. But what Doug and Sergey and Sydney and Drew have been working on is the idea that you could constrain some part of the protein to have to be functional, and then design the rest of it, and then let the rest just be freely hallucinated, whatever the network thinks would be best in the context of the fixed region. So here, for example, we have a fixed region in green that, say, binds a particular target, and then we can hallucinate everything else, and here's different solutions. So here, for example, is a central protein in the complement cascade, which is C3D. It's a big protein. It has this sort of patch here, which interacts with its receptor down here. So we can just take this part, freeze it, and then let's ask the network to hallucinate around it some sequence which has the property that it's predicted to fold to a structure, and that the green part has it has the structure that it was designed to have. It has exactly this structure. So it's sort of like a constrained optimization problem. And we get these different types of solutions out. Here's an example with insulin. So this is part of insulin here. And we can then hallucinate the rest of the protein. We can keep this part fixed, hallucinate the rest, and you can see we get different types of solutions for holding this part together. And this is work by Sidney Lazanza. This is the protein called CTLA4, which is a major cancer biomarker, and there's a protein called B7. And Sidney took this part of B7 and then hallucinated proteins around it. And so this is how we can now design proteins with new functions. And so that's what that's a long-winded answer to your question. So this was work done by Ivan and Sergey, Sam and Tamuka. Chris and Basil did the work on landscape design for landscapes, which I kind of skipped over, and Doug and Sydney and Drew are doing this constrained hallucination. So that's a long answer to that question. I promise I have no more slides.
That was great. That's super interesting. Thank you. So we do have a couple more questions here. This is from Jason Tosher, and he asks, these proteins are very interesting. For in-human use, what is the level of concern for unexpected long-term effects like cancer or question mark, question mark, question mark?
Yeah, I mean, a big question is, will they be immunogenic? And so the answer is we don't know. And we will, but as I told you, they're going into people, and we started a cancer therapeutic company, and they will be starting clinical trials on a different design protein, which is a cancer therapeutic soon. So we'll learn. So far in animals, the immune responses have been quite low, but you never know how it's going to translate person. They're quite, as far as other types of side effects, we think that they're pretty unlikely, just because we haven't really, these proteins are very specific. They only bind their target. So it's unlikely there'll be a lot of off-target interactions, but they'll have to go through they'll have to go through FDA approval, that whole set of things. So like anything, there could be always bad things, but we don't expect it, but who knows?
Sure. I guess I wonder, it seems like that's the classic. I mean, this is so cool what you're doing. I'm really impressed. But it seems like the classic science risk, right? Where you
Yeah.
say, there's this new thing and it's totally unlike anything we've ever done. And since they're completely designed and unlike nature, they're going to be fine. It seems like that's sort of the big, except for then 10 years later, you find out that you've
Yeah.
created,
Yeah.
nationality or something, right?
There's always a risk of new technology. There's no question about it. You just have to see, right?
Sure. It's neat work. I'm
Thanks.
impressed.
Thanks.
Great. Our next question is from Andrew Laszlo, and he asks, I'm curious about the protein designs that don't work. Is there anything that can be learned from them? Do they highlight any limitations or blind spots to the current design paradigm?
Yeah, that's a really good question. We are accumulating a lot of designs. I mean, we have made more designs that don't work than probably everyone else in the world put together. So so we have a lot of data on that, um, uh, and so we're starting. We're obviously trying to learn from that, and there's a little bit of kind of an irony. We have this huge amount of data, and we are doing a fair amount of deep learning now in the group, really. But we're so far where it's been successful has been learning on protein structures, like the protein structure database. Thus far, we haven't really figured out how to systematically do deep learning on the data sets we're generating. Just to give you a feel of the scale, we have data sets of multiple millions of designs that are designed to bind different targets, and we know which ones bind and which ones don't. So absolutely, it should be possible to glean something from them. And we certainly have been able to find things that correlate with binding, for example. And it's not too surprising that the more sort of specific hydrophobic contacts you have across an interface, the more likely design is to work. But I think there's a huge amount more to be done, both in sort of designing, particularly probably in designing experiments that are maximally informative for learning based on.
Great. Okay, and then we have a question from Ben Wynn. He asks or says, "Thank you very much for the talk. I'm curious about the self-assembly aspect. Does solid spherical sorry, start over here. Aside solid spherical particles and 2D structures mentioned in your talk, are there any other self-assembly morphologies currently in development in the field?"
Yeah, so Hao Shen, who recently graduated molecular engineering student, has designed a whole series of one-dimensional fibers. And these are really cool. He has many different geometries. Some of them have pores down the middle. He's trying to make conductive ones now. And we're trying to make walkers that walk along them. So one-dimensional fibers is another class. Also, very recently, Zhe, a postdoc in the lab, succeeded in designing three-dimensional crystals. So yeah, so we've pretty much made all the simple polyhedra and one-dimensional fibers, two-dimensional lattices, and three-dimensional crystals. So yeah, so it's really now at the stage of application. So if anyone has any ideas about how these things could be useful, let me know.
Awesome. All right, I think that does it for our Q&A here. Thank you again, Dr. Baker. It was so great to have you. I wish that we could give you more than a virtual round of applause here. And it's so awesome to hear all the shout outs about our MolE PhDs that are working in your lab.
Oh yeah, they're amazing. Yeah, and I encourage people to, I mean, if you want to hear more about this stuff, you'd contact me or, contact the students in my group, or, people are always excited about describing what they're doing.
Awesome. Well, thank you so much again. We really appreciate your time.
Yeah. So actually, I'll give one advertisement. So Adam Moyer, a molecular engineering student, is graduating. His thesis defense is next week, and he's been doing something completely different, which is designing using completely unnatural amino acids and backbones, designing peptides that form regular abiotic secondary structures. That some of them form these really long fibers. So it'll be a really cool talk. I highly recommend it. So yeah, thanks everybody. Thanks for your attention.
Yeah, thanks so much. All right, have a great day.
Bye.